And today it's my pleasure to welcome our first speaker of the session, Dr. Peter Robinson. He's a professor in the School of Physics and Nobel Federation Fellow at the University of Sydney. Welcome, Peter.
Thank you very much, and thank you for inviting me and giving me the opportunity to speak here. I'm going to talk about some work that's been done, as you see here, by a very large number of people, so I haven't listed all the names, on treating the brain as a physical system as a starting point for modelling rather than treating it as an AI system or a graph or something like that. So the key thing that we need to begin with is to notice that we have this physical object sitting inside our skulls, and from a physicist's side view we have all sorts of processes going on inside there, and stuff comes in, either natural stimuli or manipulations that we do to this, and we look at outputs. Historically there have been behavioural outputs, more recently there are all sorts of measurements, and we'd like to know what sort of measurement to expect if we put in a particular stimulus, perhaps one we haven't put in before, and also if we see particular measurements, what does that say about the processes that are going on to produce those measurements? How is the brain processing those signals to produce an outcome? And of course we have all sorts of different kinds of measurements, some of which I've shown some pictures from here, and a lot of us are familiar with these. They have different kinds of spatial and temporal scales. We see here that EEG has a very coarse spatial scale, but it's got very good time resolution. MRI is the other way around. For functional MRI that measures neural activity, very poor time resolution, but good spatial resolution. So if we look here, we can plot those types of measurements in a diagram that shows us the temporal scale from milliseconds up to months or even years, and spatial scales from submicron up to the whole brain. And so the problem is that we need to somehow interrelate all these measurements and phenomena. And the problem is a little bit like this one. We have all these clear measurements looking through different keyholes, and the question is, what are we looking at? How do we tie these things together? And I think I heard the answer there. I've got to come over to my. How do I click on this to? Well, I want to click on it, but I don't know how to click on the image in this system. Is there a way of doing that? Oh, okay. Thank you. So there we are. So for many of you got that. Sorry for that interruption there. But you see here we have these very clear pictures in certain sub-regimes, and we only need a very blurry picture to connect them up. Something very rough is enough to make sense of how these things interrelate. And in neuroscience, this is a little bit like people who have what one of my colleagues called Scalechauvinism. They think that their scale, be it the synapse or the single neuron or the neural assembly or a nucleus, their particular scale is the be-all and end-all of neuroscience. And so you'll have somebody comes up with the Tusk index, and they develop this in isolation. It's a great index for tusks. And then later on they meet the legologist, and who has the leg index, and they find that there's a correlation between these. And so they do ANOVAs and p-values and so forth. But it's a lot easier to realise that the tusk and the leg tend to go together when the elephant gets bigger. And so any kind of approximate model that can tie these things together can help us. I'll just move that out of the way. And so our idea here, starting from physics, is to develop integrative theories, integrative models that interrelate many kinds of observations, many kinds of phenomena, so that somebody working in one area can get an idea of how their work relates to something in another area, and hopefully we can make predictions, at least approximate predictions, of what's going on. And so our philosophy is to base our approaches on the physical system, because whatever measurement you've got, it's generated by the physical brain. There's one object sitting under there that's generating your measurement via various processes. We want to make testable quantitative predictions from ideally just one or a very few theories. We don't want a new theory every time we come to a new phenomenon. We don't get a new elephant every time we measure a different bit of the elephant. And we want to have the simplest possible approach, but no simpler, quote from Einstein. And we see from the picture there that it's possible with a pretty simple set of ingredients to get a fair approximation to what's going on. We have a much better idea of who we're dealing with there than if we had no model at all. So the integrative theory I want to talk about today is neural field theory, and this is a theory that ignores the individual neurons except in their average sense. We look at their average firing rate, their average soma potential, the average strengths of connections between neurons and so forth. And we encapsulate in an average sense things like the rate at which spikes are arriving at the neuron phi, the cell body potential that's generated on average in a population of neurons, V, the firing rate with a threshold response, Q, and then the outgoing field of spikes, which then forms the input to the next lot of neurons. So we get a cyclic set of equations. I'm not going to write down the equations. I only have about 10 or a dozen variables in the models that I'm using today. So I think I'm the tail end Charlie on the number of variables here. And three differential equations for each population of neurons. And so we get a closed set of nonlinear equations for these quantities. But if we're dealing with a physical system and we want to make testable predictions, we ought to make some comparisons with data. And so I've just come up with a few examples of the sort of comparisons that we've done, because this is really, in this talk, not focused on making predictions for one single thing, but showing that you can have a theory that will make some kind of predictions across a wide range of different phenomena in the hope of interrelating things. So first thing here is we've got some time series, and the right hand are experimental time series. They're taken from Nunez's book, but it's an even older set from one of the early EEG textbooks, and the name escapes me. You can see it's very early because if you look at the large amplitude alpha oscillations in eyes closed waking, you see they're slightly curved because they've been drawn with a pen on a roll of chart recorder paper. So that's really old. That's, I think, from about the 1940s or 50s, that figure. And at the left column, so we have eyes open, eyes closed. We have sleep stage 2 with some spindles, 15 hertz spindles, and we have deep sleep with these slow waves, these up and down states. And on the left, we have our simulated versions of those time series, and you see there's a lot of similarity. I haven't tried to match the individual cases exactly there, but there's a lot of similarity in the frequencies and the relative amplitudes. And that's recorded with EEG. If we look in more detail at the EEG, we see the EEG spectrum, power versus frequency, and we see a good match between the dotted line, which is the model, and the solid line, which is experimental data from one subject. And that was one that didn't have much muscle artifact, which would produce a tail out here. We see a very good match, and we've fitted the 10 or so parameters of the theory to these data. If we're going to have 10 or so parameters, we'd better compare with more than one kind of data. So we looked at evoked response potentials, and again we get a good match for the similar parameters. And we can explain a lot of these features in terms of resonant frequencies and other features of the physics. So we see the alpha rhythm. It turns out that that's due to a resonance in signals passing from the cortex down to the thalamus and back again. And the beta frequency is not a separate thing. It's the harmonic of the alpha in this model. And most of these phenomena we're modeling as arising from the corticothalamic system, which generates most of EEG because the cortex is close to the skull. Now, if we can fit the parameters to the data, we can estimate the parameters from the data going in the reverse direction from data to model. And here's a map, for example, of the time delay for signals to travel from the cortex down to the thalamus and back again. And we see it's a little shorter on average towards the back of the head. Here's the nose and the ears and the circles of the EEG electrodes. And that fits with the fact that the alpha frequency is known to be a bit higher at the back of the head. And we can also do studies versus age. This is from age 6 to 86, 1,500 subjects, estimating that time delay. And we see the time delay falls in young people up to about their late teens and then gradually rises back again. We interpret that as being consistent with myelination of the corticothalamic axons in childhood and teenage years, followed by perhaps cumulative axonal damage or disease that causes the conduction velocities to go down again in old age. So that fits with physiology. So we can link to things like electrophysiology, physiology, anatomy. We can also look at different states. So in this example, we've got states of arousal. Here are some data, and these are from different states: eyes open, eyes closed, waking, REM sleep, sleep stage 1, 2, slow wave sleep, which is the deepest, and spindle state, which is when you generate those 15 hertz oscillations. And we see here in the simulations or in the calculations that we can match these features pretty well. In waking, we have the 10 hertz alpha. In sleep stage 2, we have the 14 or 15 hertz sleep spindles. We also see the beta in the waking state. And in deep sleep stages, we have this big enhancement of low frequency delta waves, which are observed. So we can use these features to automatically track the brain state and some physiological parameters that underlie the model in real time. And so here, I don't have time to show the movie, but here we have a diagram that shows basically the strength with which the cortex is listening to the outside world, and this is the strength with which the cortex is listening to itself through self-feedback. And we start here in eyes closed, near to this boundary, which is a critical boundary beyond which we get an instability, a seizure. We start quite close to criticality, and we drop vertically downwards, more or less, with a few wiggles, and end up here where the brain's no longer listening to the outside world. This is falling asleep. That's sleep stage 1, the transitional state, excuse me, in going into sleep. And then over a couple of minutes, this strength increases and we move across here into sleep stage 2. Again, we're near the critical boundary. We get this very strong critical enhancement of power at low frequencies, and the cortex is listening to itself. So we've cut off the outside world, we're in our own internal world, and we can map that quite reliably in real time. You see there's quite a bit of jitter because of noise in the signal and possibly fluctuations in the state, but nonetheless the general trend is clear. What happens if we go across this boundary? Same model, remember. If we go across the boundary, we end up in a seizure. And here's an example of an absence seizure, which is quite common in small children. A good fraction of 1% of children have these. And what we see here is a pre-seizure state. Then we get 3 hertz spike and wave oscillations, and this is the frequency versus time, and we see the 3 hertz switching on very, very strongly, and a whole bunch of harmonics during the seizure, and then at the end of the seizure it switches off and we come back to more or less the original state. Now the left-hand column is actually the model, and the right-hand column is data from a child at the local hospital, and we see it's not quite as even, it's a little bit more ragged, but again we have a 3 hertz and we see it and a whole bunch of harmonics. So the same model can look at abnormal states such as epilepsy and interrelate them as being beyond this boundary, interrelate them with the normal states and map those normal states within the space. And the last thing I want to talk about is just a flavour of where the field is going. A lot of recent work in EEG and particularly in functional magnetic resonance imaging has shown that there are patterns that show up over and over again in the activity of the brain under both resting state and task-based states, and these are called resting state networks. I think networks perhaps a misnomer, but these are actually not maps of activity. They're maps of the of a thresholded statistical significance measure of the difference between a task and a baseline, typically. So don't mistake these for activity maps, but they're characteristic. And at the moment, we find that calculating from our theory, we can calculate patterns of activity, natural modes, like the notes of a violin string. And we can look at the spatial patterns of these. And the lowest one, the lowest frequency one, is uniform, where the whole activity in the whole brain goes up and down together. This is actually just one hemisphere, two views. Excuse me, flattened out, it looks like so. And then I'll put that down there and get my speaker away, my microphone away from it. And then we have these modes where half the activity is up, half is down, and it oscillates back and forth. And the current question is how this relates to these modes. And I'll just stop there with the summary. Sorry about that. So we're still looking at that, and that's a current hot topic of interest in the field, is how the patterns of activity, why are they so stable? And that's probably just in the same way that modes on a violin string are always the same. They're set by the geometry of the state. So I'll stop there. Thank you very much.
We have time for a number of questions. Just raise your hand and we can have someone find you with a microphone. Christoph.
Peter, how good are these models at fitting individual subjects? I mean, not just averages, but you take me, for instance, when I sleep or happen to have an epileptic seizure, etc. And and then how difficult is it to model Anton's brain? Not in the aggregate, but at the level of individual.
Sure. Well, that state tracking was done for an individual subject. We do that quite regularly. We also fit the parameters for individual subjects. So you saw that 1,500 subject scatter plot. Each one was fitted individually. Now, there's obviously more uncertainty in the parameters if you fit just one person or you fit a very short bit of data, but than if you fit in the average, but you can.
How many degrees of freedom do you have per?
There's about 10 parameters that we fit. And all of these parameters are constrained by the physiology, by independent measurements. So none of them are completely free parameters. We can't just adjust them at will. We have to stay within the physiologically realistic ranges.
The microphone's coming to you right now. Right there.
You referred to neural field theory. What kind of field are you referring to? Are you referring to electric fields, magnetic fields, or electromagnetic fields?
Oh, that's a good question. Well, we have several fields that we look at, and they're basically, we average quantities like the firing rate of the neurons, the average soma potential in the neurons, and so forth. And each of these has a local average value at each point, which varies from place to place. And that's the field. So these fields are things like voltage, firing rate, and so forth. They're not electromagnetic fields in themselves, but you can compute measurable quantities from them. So, for example, if a neuron is firing at a certain rate, it'll produce a certain amount of electric dipole around that neuron, and that adds up to produce the EEG, for example.
And the last question?
Yeah, I have a question jumping off of that. How well do you think that your neural field theory is actually approximating what an astrocyte does in the physical brain?
An astrocyte?
Yes.
Well, we're mainly looking at neurons, but we have modeled astrocytes as well using similar methods. And again, we're averaging over populations of cells. So in the average, it seems to be a pretty good approximation if we look at the observations. The ultimate test is the observations and whether we can reproduce them, and whether one theory can reproduce a whole range of observations rather than just one or two.
Okay.
Okay, thank you.
Thank you so much.